Yeah, so we'll start today's class with a revision of meiosis because I want you to go through this revision compulsorily because otherwise you might get confused. Okay, so what did we saw in yesterday's upload? So at the end of meiosis I, how many cells were formed? Do you remember? We had a parent diploid cell, isn't it? We had a parent diploid cell which was 2n, right? And this 2n undergoes the process of meiosis I, giving you what was the result? 2 haploid cells. Okay, 2 haploid cells, and because it is haploid, it is just 1. So 2n becomes n. The result from one parent cell, you get two daughter cells with haploid number of chromosomes. So now I need to ask you, are these cells ready to become gametes? Are these cells ready to become gametes? So can you say these are gametes? Answer is no. Okay, answer is no. Now why? Because each chromosome, alright, each chromosome, the answer is no, because each chromosome still contains the sister chromatids. Okay, each chromosome still contains the sister chromatids. Yesterday, if you have remembered the process, you saw that at anaphase there was no splitting of the centromere and one chromosome just moved into the opposite pole, isn't it? Now what do you mean by this one chromosome? If I draw this chromosome in detail, you will see this is a haploid chromosome. Okay, this is a haploid chromosome, isn't it? But with a sister chromatids. Okay, so these two are the chromatids. So chromatids mean the chromosome is present here, right? The chromosomes are present here. Okay, so this is a detail or else explain, the magnified view of this chromosome, isn't it? This was the thread like diagram which was used to explain you, but actually this is what. So you have two chromatids, isn't it? The two chromatids. Now you might have seen in mitosis, this one chromosome when undergoes mitosis, the centromere splits and each pole receives the sister chromatids, isn't it? Each pole receives the sister chromatids, isn't it? So this is what has to happen in meiosis II. Okay, so in meiosis I, these are not gametes because it still contains two sister chromatids. So you have to actually make it more, you have to reduce the cell into such a way that each cell contains only one sister chromatids. Only then it becomes a gametic cells. Hope it's clear, students. If not, please do comment me so that I can answer you in a better way. If not, okay. So therefore we need to have the process called as meiosis. Okay, so meiosis II is definitely required. So this meiosis II is also called as equational division. Equational division. Meiosis is also called as equational division. Why? Because at the start, okay, so what is going to undergo meiosis II? Now these two cells are undergoing meiosis II, isn't it? These two number of cells, each having one n. Okay, so at the start, how many chromosomes are there? So at the start there is n chromosome, isn't it? So what happens at the end? At the end of meiosis II, you will have the same number of chromosomes. So n number of chromosome, n number of chromosome, and this is why it is called as an equational division. Hope it's clear. Okay. So actually the chromosome number does not change. Okay, the chromosome number does not change. So what is actually changing? It's only the DNA content which is getting changed. So chromosome number is the same, isn't it? Now we'll try to explain it in a more better way. So meiosis I, also called as reduction division where the parent had two n number of chromosome, one from a father and one from a mother. So after the meiosis I was over, each cell received only one pair of chromosome. Okay, now this has to undergo meiosis II. So meiosis II, this cell has to undergo meiosis II. So what happens in meiosis II? This undergoes meiosis II. So the N becomes N. Okay, and this is why it is called as an equational division. Hope it's clear. Equational division. Okay, so let us see the stages of meiosis II. Meiosis I was over yesterday. So stages of meiosis II. So as we have seen, in every mitotic and meiosis you have prophase, you have followed by prophase, you have metaphase, then you have anaphase, and finally you have telophase. Okay, so this is common in all mitosis as well as meiosis I and meiosis II. So how do we differentiate? By just giving the Roman number 2. Prophase II, metaphase II, anaphase II, and telophase II. Okay, so these stages are, or else they actually closely resemble the mitosis process. They closely resemble the mitosis process. So today's class will not be much confusing and just have to follow mitosis. Okay, so we'll start with the first phase, which is prophase II. So what are the events which is happening in prophase II? Now here each haploid. Okay, so you have got how many haploid number of cells? 2 cells, isn't it? You had 2 cells at the final result of meiosis I. So now each haploid, that is 2 cells, each of them, okay, this will enter into prophase II. Okay, so separately each cell will enter into prophase II. So what happens in prophase II? The chromosome condenses, condensation happens. Okay, chromosome condenses. Okay, nuclear membrane, NMS, nuclear membrane, as you all know, nuclear membrane disappears, and then there is a nucleolus which also disappears. Okay, and finally there is a spindle fiber formation. Okay, so there is finally the spindle fiber formation which happens. Okay, so one of the neat question which is possibly asked is, do the DNA replication occurs before prophase II? The answer is no. Okay, there is no replication of DNA molecules before prophase II. So I mean to say, there is no involvement of S phase which happens in meiosis I. Okay, so this S phase, which is a part of a interphase where the DNA duplication happens, DNA replicates, isn't it, giving you the double the quantity of DNA, if you remember. Okay, so 46 chromosomes and then 92 chromosomes, isn't it, right? If you go back to mitosis I have explained there what happens in S phase of interphase. So in prophase II there is no S phase happening, and hence there is no DNA replication. There is no DNA replication. So, DNA replication does not happen in Prophase II, a very important fact which usually is asked in the NEET questions. Okay, so what is happening in Prophase 1, Prophase 2? So, simple, the chromosome condensation happens. So, we have two chromosomes, one is this and the other one is this, isn't it? So, this condense and there is a disappearance of nuclear membrane. So, because it is condensing, you can visibly see it, disappearance of nuclear membrane and followed by this is your cell membrane. Okay, and also there is the formation of spindle fibers. Spindle fiber formation starts happening at the Prophase II. Okay, so these are the events which you will see in Prophase II. Okay, so let us move on to Metaphase II now. So what happens in Metaphase II? This is your Metaphase II. Now you should all be saying it, what is happening in metaphase. All of you know that the chromosome arrange in a equatorial plate. Chromosomes arrange in the equatorial plate, isn't it? Yeah. Equatorial plate, right? So, chromosome arranged at the equatorial plate and also the spindle fiber attached to centromeres. Attached to centromere. Okay? Spindle fibers attached to centromere. So, if you draw this, how will you see this diagram? So, I will draw this like this now. Okay? So, because the diagram is not much complicated, because of there is no crossing over and all that. So, I will draw it in this way as you have seen in your textbooks. So, these are the two chromosomes, okay, right, and the spindle fiber attached to the centromere. The nuclear membrane has already disappeared and hence there is only the cell membrane, okay, right. So, the nuclear membrane has already disappeared. So, these centromere are attached to the spindle fibers now. Now, this is in Metaphase 2, right. So, what is the difference between Metaphase 1 and Metaphase 2? Now, if you ask me, in Metaphase 1 what was happening? In Metaphase 1, that means what process? Meiosis I, isn't it? What was happening? The analogous chromosome pairing was happening. Sorry, homologous. Homologous chromosomes were aligned at the equator, isn't it? Okay, if you remember. Now, what is happening in Metaphase 2? In Metaphase 2 or else in meiosis, you can also say this process as Meiosis II, Metaphase Metaphase. So, what is happening here? Here the individual chromosomes are arranging at the equator of the cell, okay. So, this is the difference in Metaphase of Meiosis II, individual chromosome arrange, and Metaphase of Meiosis I, homologous pairs arrange. Homologous pairs means one from the father and one from the mother. Now, here we do not have a pair, we have just one individual chromosome. So, after shuffling, after the Meiosis I, this is one haploid cell, isn't it? One one cell having the n number of chromosome, and this is now going to undergo the process of division, meiosis process. Clear? Okay. So, this is what is happening in your Metaphase 2. Okay. Now, we will move on to Anaphase II. Anaphase II. What happens in Anaphase? Remember? If you remember, the centromere splits, okay. Centromere splits. Okay, sister chromatids separate. Sister chromatids separate. Okay, and then because of this, what happens? They move towards the opposite pole. Move towards the opposite pole. Okay, opposite poles. So this is what happens in Anaphase II. Now again we'll try and compare what happens in Anaphase I and Anaphase II. Okay, very important. In Anaphase I or else Meiosis I of Anaphase, what happens? There is no centromere splits. Okay, there is no splitting of the centromere. And what is happening in Anaphase II of Meiosis II? What is happening here? The centromere splits, isn't it? Now because of this process, what is actually the difference? This is where we actually see the difference. There is no centromere splitting. So homologous chromosome which was present like this, one from parent and one from father, one from mother. Okay, so these chromosomes were separated. So individual chromosomes were students, please zoom in when I'm drawing in so small structure. Try zoom in and see what I'm trying to say. So here you can see the individual chromosome is getting separated. Now here what is going to happen? Here the centromere will be split. Okay, so giving you the structure like this. So the sister chromatids will be separated. Okay, here there was no sister chromatid which was separated. Here now the sister chromatid will be separated. Hope it's clear. Okay, so let us try and draw the diagram now. So here you have one chromosome like this, one sister chromatid and the other sister chromatid moving towards the other direction. Okay, so the chromosome with two sister chromatids now separated, okay, because of the splitting of the centromere. Okay, so yes, so this is the cell membrane now. Okay, so here you can see from this stage, okay, the cell is now becoming like this. So the chromatids, sister chromatids are now separated, right? Okay, so moving towards this direction and this is moving towards this pole, right? Okay, so this is followed by what stage? If you know, after anaphase, the final stage is telophase, telophase II. So here what is happening? What are the special events which is happening? So here the chromosome reach the pole, okay, chromosome reach the pole, irrespective opposite poles. Okay, there is nuclear membrane which reappears. Nuclear membrane reappears. What else has to reappear? Nucleolus has to reappear. Okay, nucleolus has to reappear, isn't it? Okay, so here if you draw the diagram, you will see the nuclear cell is there. I'll draw it here. Cell membrane is there. Okay, the nuclear membrane has reappeared and the nuclear membrane reappears around where? Around the chromosome, and chromosome has already reached the corner, okay, the pole opposite end. So this is one chromatid, this is one chromatid which is reached the pole, and this one is the another one. Okay, so here it has reached to the poles now, and now this is called your telophase II, right? So here each cell has received the same number of chromosome. The chromosome number is same. Okay. So, the chromosome number is same because the sister chromatids has separated, isn't it? The sister chromatids has separated. So, N is equal to N. So, this is why it is called as the equation division. Clear? Okay. So, after telophase what happens? The stages cytokinesis. Okay. So, cytokinesis, as all of you know, cytokinesis is what? The division of cytoplasm. Cytoplasm occurs. Okay. So, division of cytoplasm. So, finally, after the division of cytoplasm, if you see, the cell will be like this, isn't it? So, one cell, so cytokinesis is happening, you get the two cells because of cytokinesis. You know what is cytokinesis? The cleavage furrow formation happens, this deepens, causing the formation of two daughter cells, isn't it? So, cell membrane, nuclear membrane, and then each one receiving one chromosome, isn't it? Each one receiving one chromosome, and the result is N and N. Okay. So, two daughter cell. This is your two daughter cells. Two daughter cells. Right. Now, this is the result of one haploid cell. Okay. How many haploid cell did we get after the meiosis I? You got 2, right? So, this is the result of one haploid cell, and the same thing will happen with the second haploid cell. So, your second haploid cell will also result in the formation of two daughter cells like this. Yes or no? Right? So, finally, after the meiosis II is completed, you get how many daughter cell? You get 4 daughter cells, 2 from each parent cells. You get 4 final daughter cells, 2 from each parent cell, isn't it? Okay. So, what is the significance of this meiosis II finally? The significance is that the sister chromatid gets separated, okay, and completes the gamete formation. So, sister chromatid gets separated, and this in turn causes the gamete formation, because here the sister chromatid is separated, giving you the haploid number of chromosome and not diploid. Okay. So, in meiosis I, what did we see? The result was N, but then the chromosome sister chromatids were together. So, you cannot say it is haploid. Okay. So, here actually the chromosome has reduced, giving you the haploid, because the separation of the sister chromatid has happened. Okay. So, this is your finally, if I conclude, one parent cell, okay, one parent cell, right. So, undergoes meiosis I. Okay, I am writing MIS, meiosis I gives you 2 daughter cells, right, and this 2 daughter cells each undergoes meiosis II, right, giving you 4 haploid cells. 4 haploid cells, because the sister chromatid has separated. So, this is the conclusion of meiosis II. Hope the class was clear, students. Do comment me if you have any doubts, and do subscribe, share and like the channel, and this really will motivate me to bring more classes for you. Thank you.